Welcome to Global Healthcast, brought to you by Global Health Press. This is a very special edition on pandemic preparedness, and we have the honor to have with us today Professor Bekeredjian-Ding. Good morning, Professor Bekeredjian-Ding.
Hello, good morning, everyone. Thank you for inviting me. I'm very glad to be able to discuss pandemic preparedness with you today.
Professor Bekeredjian is an MD PhD with a broad international educational background in medical microbiology and immunology. She's currently working at the German Vaccines and Sera Licensure Authority, the Paul-Ehrlich Institute, or PAI, in Langen, Germany, as head of the Center for Pandemic Vaccines and Therapeutics. Her past offices included membership at the CHMP Vaccines Working Party at the European Medicines Agency, and as some highlights only on her current memberships, she is a member of the WHO Product Development for Vaccines Advisory Committee, or PDVAC, and a member of HERA Advisory Forum. Professor Bekeredjian-Ding, it is a great pleasure and honor for me to have you with us here today, and we're looking forward to your presentation. And if you don't mind, in between, I may interrupt you to ask some stupid questions. Thank you very much again.
So thanks a lot again for the invitation. I think I'd like to start out by explaining about pandemic prevention and preparedness. Both are important, and I think we learned with the COVID-19 crisis that there is many things we would like to prevent in the future. So there was health issues, high mortality, long COVID syndromes. There is economical debts, bankruptcy, investment losses that we would like to circumvent in the next pandemic. And of course, we also saw there was supply disruption, there was export bans, and a lot of happenings that were really leading to failure and disaster in our populations, and this worldwide. And of course, there's now many initiatives to try to see how we can prevent this in the future, but I personally believe that every state, every government has to seek some way of preventing this next time, because we know that there's going to be more and more pandemics. Now, what is needed? What is this prevention and this preparedness? I think, first of all, we're going to need early warning systems, and WHO is taking a stake at bringing all the players together here. Then we have to be very aware on a regional level what the infection control measures are, what diagnostics we might need, and how to develop these diagnostics as fast as possible. And then, of course, we are in any case going to need either vaccines or therapeutics or both. So this is really the task that I'm involved in. And I think it's very important to explain to you right at the beginning that there is some known pathogens that are dangerous and that we are afraid of spreading throughout the world, but there's also a lot of unknown pathogens. And we need to be prepared for both. Now, the production and distribution of vaccines and pharmaceuticals is then the next step. So we would develop vaccines and therapeutics, manufacture them, and then have to distribute them. And in this COVID-19 crisis, we also learned that that is not something we should take for granted. It can be more complex through, well, special needs for the transport, like the -80 for the COVID-19 vaccines. Or there is also other possibilities that can happen, circumstances we might not be able to foresee today. And so this means we need to have robust but very flexible systems at the same time to enable us to bring vaccines and therapeutics to any place within our countries and the world. Now, we'd like to start with the short risk evaluation. I think it is important that for any pathogen that we see, there can be a pandemic potential. We need to evaluate this risk. And this would, of course, go along with high infectivity and contagiousness with and without therapy or vaccination. Now, when we think about preparatory measures, these have to involve known pathogens with high lethality, let's say Ebola or Lassa, for example. There is a disease risk that is due to high virulence and high pathogenicity of these agents. And now what can we do to find this? There is, of course, epidemiological observational studies, but this kind of study networks has to be built, it has to exist, it has to be funded over a long time. Animal studies, they are important to understand whether there's transmissibility to humans, for example, but it's also important to understand whether an animal model that we might have available for development of drugs would even be predictive of what happens in humans. There's been a lot of discussion in COVID-19 about human exposure studies. Human exposure studies have some value at the beginning of a crisis, of a pandemic crisis, but there are also limited in the number of participants and subjects that will be able to be included. And we always will have to very well consider whether this is an option really to accelerate development of drugs and vaccines. But it is definitely something that the WHO is supporting and that might help us in the future if we are, again, prepared, if we know how to do this, how to conduct this. And then, of course, there is the stake for in vitro models. This is animal models where we could test, of course, in vaccines and therapeutics in the animal, in a live infection model. But then again, we try to substitute this by mucosal models, by other types of in vitro and in silico studies that could maybe be faster and replace animal studies to a certain extent. So this is an area where I think we need to have research. The next step basically is development of drugs and vaccines for pathogens that we already know, and eventually even a stockpiling. If we know there is regular epidemics, it might be worth to stockpile certain amounts that might be needed just to be able to react in an emergency. Further, if we know the pathogens, we can of course already start to think about what are the specific protective measures. And we're, of course, clear this depends on the infection route. So if you have respiratory infection, you might benefit from masks and so on, and what is really going to be needed to protect the population. Finally, what is important to be observed is that if we don't foster and fund development of therapeutics and vaccines in this area, we have a valley of death. If there's no cases, why should somebody produce something? But what happens when the cases show up and we have nothing available? So this is where we get into the need for clinical studies, for proof of efficacy and safety that cannot be provided, and thus we cannot authorize this kind of medicines. But we need to think on how we could take the development forward to a point that these studies could take place as soon as cases are available and there is an emergency where these drugs are being needed, and how to then do this very, very fast. Secondly, there is known pathogens with development of resistance, for example, against antibiotics. So here the danger is we know these pathogens very well. We treat them every day, but we don't know when they become resistant, whether we will be able to treat them in the future. This is basically most of the field of AMR, and there is special measures being taken to try to tackle this on a global level, but it is probably something that we would call a silent pandemic and something we will need to be prepared for. I will show some examples later why this can also impact pandemic preparedness. Finally, we have the unknown pathogens. This is something the Center for Pandemic Vaccines and Therapeutics at Paul-Ehrlich-Institut deals with. We basically try to be agnostic to the pathogen, but to be prepared for dealing with new drug developments. And I will be relating to this later. Now, when we look at the risk factors for pandemic spread of infectious agents, I think we're all aware that poor sanitary conditions are very important and are mostly the reason for epidemics that we observe worldwide. COVID-19 was a good example where we had animal-human contact. So these are zoonoses that bear a very important danger for pandemics. We also know this about avian influenza and other diseases that we've seen in the past. There is also the risk of development of antibiotic resistance through exposure to antimicrobial substances. I'll show you some examples there. And of course, the most important factor is probably globalization, because we travel. There's no frontiers for infectious agents. And so we carry these agents with us when we pass from continent to continent. And this is, of course, increasing the risk that an epidemic turns into a pandemic. Finally, we're experiencing a climate change. There's water warming, there's proliferation of vectors, ice melt, and I think that's That's also a very important factor. Now, this is looking into the past. You see here that we used to have a lot of bacterial infections as pandemics. We have been able to constrain this, to control this through hygiene measures. This has been very important, and it's really a big step forward that we have been able to secure water quality, for example. But we do see that there is outbreaks anytime we have floods, we have earthquakes, and similar situations of war that then endanger water quality, endanger the hygiene conditions, and therefore lead again to new epidemics. So this is still something that happens nowadays, just not in this extent. In the last decades, it has mainly been viruses that have seemed to be a threat. You remember the Spanish flu a long time ago. You remember the SARS, the first SARS epidemic. And of course, we have been lucky that COVID-19 was not as lethal as we first expected, but it has had a lot of impact. And again, it's a zoonosis. And so this is an area where we have to look into more closely. Now, when we focus on what has happened in the past and what could happen today, I think it is very important to think about antimicrobial resistance, because cholera or Shigella or other pathogens that can be transmitted very easily and can become dangerous are becoming more and more resistant. We see here that there is a Shigella spread worldwide that is basically associated with multi-drug resistant Shigella. We see here also that there is contaminated water sinks with Shigella and Salmonella that can become a danger. But one other aspect is, of course, also TB. Tuberculosis is a disease that is latent in many individuals that were infected and then breaks out at some point when we see a weakening of the immune system. And this type of infection is dangerous for us because we don't see it that easily. And here we see that 1 in 3 deaths in TB come from antimicrobial resistance. And the fact is, we know how to treat TB, but if the bacteria are resistant against these antibiotics, we need about 2 to 4 weeks to find the right antibiotics and to find out that these bacteria are actually resistant. And so we need to change the scheme of antibiotics we use for treatment. And this takes time because the bacteria grow very slowly, but it bears the danger that during this period, this person is still infectious. And it bears the danger that this type of drug-resistant bacteria are being passed on to others. Now, we thought for a long time that we would no longer see Yersinia pestis. Now, looking back in the 6th century, we had a spread from Egypt to Europe for 50 years. Then in 1347, these bacteria spread from Central Asia to the Crimea and to Europe. 25% of the population died. So we all remember the Middle Ages and know that this was really a severe event causing lots and lots of deaths. Now, when we think about plague, we do not need to think about medieval ages. In 2017, we saw an outbreak in Madagascar, and we see that there is more and more infections, and that the World Health Organization is highly involved in trying to resolve the situation. Now, what can be done? We fight the plague with antibiotics. Here there was import of antibiotics into Madagascar, and preparedness in this case also meant to be aware of which countries could also be affected. But I want to highlight here that during this outbreak, there was also observations that there was transmission of antimicrobial resistance through Yersinia pestis pathogens, bearing an additional danger that those antibiotics might not be fully functional for what we thought. Now, very similar with cholera epidemics, we've been seeing more and more epidemics in all kinds of cases of crisis right now, most importantly in Ukraine, of course, where there's war, in Syria and in Lebanon. Many cases, some deaths. And despite our very highly developed clinical medical healthcare systems, it is still a lethal disease. Now here we do have vaccines, and we can decide whether to do ring vaccinations or whether to do mass vaccinations. There's different advantages to both, and this needs to be discussed in the situations. But it is definitely something that is possible because we have vaccines available. Now, again, the WHO here has a stock for cholera vaccines that can be used for emergencies. So here we have an example how this can be prepared. And we see that with 2 different vaccines available, of course, we have more availability of vaccines. But despite of this, we've seen shortages. And this has, of course, also had consequences for the epidemics we're seeing in Africa right now. And again, I'd like to highlight there is antimicrobial resistance in cholera. These pathogens are also acquiring resistance. And this also defines the need for intervention and also the advantages of using vaccines to prevent this disease. Maybe I should, before I go over to the viruses, ask now whether there would be some points we could discuss.
I think I'm well with you, and maybe I ask some questions in the end on resistance and some other topics that just come to my mind while you're speaking.
Okay, fine. So now I'd like to turn to the viruses. Here, there is a list. There is a whole lots of lists, but they basically are all very similar with viruses with pandemic potential. So here's the SARS, coronavirus 1 and 2, Nipah, MERS, Ebola, you name them. Now, there's been a very important project called PREDICT in the past that identified new viruses. They looked at new coronaviruses in Latin America, Africa, and Asia, but also others. And most of these viruses were actually of zoonotic origin. The WHO has defined priority threats or diseases for R&D, and these are the viral threats here. COVID-19, of course, continues to be a threat throughout the world, although it has become endemic in many countries. There is Crimean Congo, hemorrhagic fever, Ebola viruses and Marburg virus, Lassa fever, the MERS virus, which is the Middle East Respiratory Syndrome coronavirus, and SARS, also both of zoonotic origin, Nipah, henipaviral diseases, Rift Valley fever, and Zika. And what today is defined as disease X. Disease X is basically the unknown pathogen I was talking about at the beginning. And it is supposed to make us aware that we need to have technologies available that are so versatile that we can develop vaccines and therapeutics within a very short time frame to an infectious agent we do not know today. And so this is basically what the WHO recommends to work on, and it will help then funding agencies in all different regions of the world to align a little bit on where it's worth and where to prioritize their funding in views of pandemics. Now, when we talk about vector-borne disease, one virus that comes to mind is West Nile virus. It is found in birds and Culex mosquitoes and transmitted easily. And here we're already seeing some influence of the climate change, because we see that it's increasing within the EU, within Germany, which is a northern country. And we see that, of course, some preventive measures that were already recommended by the CDC might soon be necessary. And I've been involved in this at the Paul-Ehrlich Institute because we had to change our regulations for blood products, actually, to look into West Nile virus, to detect West Nile virus in blood products, but also, of course, to build up a surveillance within Germany. And try to understand how many human infections there are and how the representation within the animal population is. Now, if we continue with climate change, another type of bacteria that has been emphasized is Vibrio bacteria. They're basically similar to cholera, related to cholera, but we call them Vibrios of non-cholerogenic origin, and there's different species. They live in the environment usually. They have very different nutritional conditions that they prefer, and you very often have either wound infections or infections via direct contact, so via oral ingestion, for example. And then this would lead to diarrhea if it's via food, or if it can lead to persisting wound infections that are just not healing. And person-to-person transmission is actually relatively low, but the problem is really that if they can grow better in the environment, or for example, what we see in Germany is in the North Sea, then of course the incidence increases. We finally are seeing that there is a revitalization of the permafrost, that there is ice melts, and we were surprised with the finding basically that this means that we're basically also revitalizing infectious agents. This includes smallpox, Bacillus anthracis, that basically also has led to infections in Siberia, influenza viruses and Acanthamoeba, and many other viruses, some species that are not that well known, but it does show us that there is a potential source here of finding new areas of spread of long forgotten agents. Now, if we think about what the pathogen-independent preparatory measures would be, I think the most important thing is to focus on manufacturing and distribution. Both were bottlenecks of the COVID-19 crisis, because you can develop the nicest vaccine. If you can't produce it on a high scale and you can't distribute it, it will not reach the people who are supposed to be protected. And so this is something that was a real challenge in COVID-19, and you see here, how many different types of initiatives showed up to solve this problem of fast access to pandemic vaccines. You see in the US, everybody is very well informed about Operation Warp Speed. In Europe, we have the HERA incubator and now the foundation of the HERA at the European Commission to take care of these emergency situations. We have a lot of funding going in here with COVAX distributing vaccines and the ACT accelerator to make sure that there is equity and equitable access throughout the globe. But we've also seen this is not easy. Now at CEPI, we have the vision that we want to limit pandemic losses by creating a framework, which is infrastructure, technological and digital infrastructure for making pandemic vaccines available as quickly as possible. This is in 3 to 6 months, basically, is the goal. It's very ambitious though. And we are focusing not on the development, because the development would be funded by research agencies mainly. This is sustaining promising vaccine platforms, emerging technologies for vaccine delivery, and networks for preclinical and clinical development. And so knowing that this will happen, basically, we start a little bit later on trying to secure the manufacturing capacity within Germany, and we work together with HERA at the European level to do so. So basically, our aim is to map the manufacturing capacities, to secure them, to be prepared for production and filling in the case of an emergency, to stabilize also the supply chains, because you can only manufacture when you have raw materials and consumables available that you need, and to secure scalability so that there is a rapid increase in mass production, basically, of vaccines for mass vaccination. Finally, distribution of vaccines includes a toolbox for distribution, which is of course very much dependent on the region and the infrastructure already set up in the region. And so I think this is really a task for every different country and region to set this up. And we are looking into this for Germany. Right now, we're still distributing the COVID-19 vaccines and storing the COVID-19 therapeutics. But of course, we're also developing new concepts, how this could be done easier, better, and so on in the future, and how we can monitor distribution, and of course, the administration and support pharmacovigilance and epidemiology. Now, with this, I'm at the end, and I think now is maybe time to set some questions.
Yeah, thank you very much for this brilliant presentation, which covered a lot of different fields. I'm really amazed about the broad thinking behind this. I have some detailed question and some more overarching question. One of the detailed question would be, if you have no pandemic, it is difficult, if not impossible, to do a study with a therapeutic if there is no exposure, right? You cannot test a vaccine if there is no pathogen around at the time. It is an ethical question. Do you think ethical review boards would approve studies with a vaccine or an agent for an organism that is not circulating?
Yes, this is actually the dilemma I was trying to explain, that we know we will need this kind of vaccines at some time point, therapeutics, but there is no clear idea how to make it available. And then, of course, companies lose interest because they don't know when it will be needed, so when they will make their profit. And it is for the regulatory agency, the data package would definitely not include studies that would show efficacy of these vaccines. And so we need to come to an idea on how to leverage this. And the most likely is, first of all, to secure that we know as much as possible about the safety of these vaccines and therapeutics. And then we need to find a common interest from public and private side to eventually stockpile the vaccine or the therapeutic till the moment comes when there's a new epidemic. It is feasible, but still difficult for infections like Zika or chikungunya, where we know that there is regular outbreaks. We just have to wait, but this can take years. And we have to come up with a concept also how we might use our preparedness facilities to produce in exactly that moment instead of having to stockpile.
A question that is then following this one, right? It's difficult to do studies if there is no pathogen. The question then is on safety. Usually good safety data is based on some 10,000 exposures to the vaccine. And again, how can you do this if there is no disease around? And I think you need to be ready with a good surveillance system in case of a pandemic only, but you cannot do it beforehand. Do you share that view, or what is your view on this?
It is extremely difficult. The only way around it is basically what is now called the platform technologies, that you have safety data packages using exactly the same kind of standardized manufacturing, but just with different vaccine antigens, and that you collect this data and you try to extrapolate what the safety risk is. But you still have to be aware that the exchange of the antigen, for example, in a viral vector, can change immunogenicity, can change reactogenicity. And so you will still have to look at the individual vaccine product once it is ready to be used, and you will need clinical trials. And I think it depends very strongly also on the perceived risk or danger situation, on how you can deal with starting or rolling out the clinical studies. But you need to be sort of prepared for clinical development in that moment.
Now, one thing you mentioned was secure manufacturing capacity. I guess this is not possible for all countries and not even for all countries in Europe. Luxembourg, with 600,000 people, they will have a hard time getting manufacturers to be to be producing in their city, right? And the same may be true for Liechtenstein and Monaco, and I mention now all the small countries, right? But even other countries still have no production capacity at home. So how would that work on a European scale? Any ideas on this?
Well, I think on the European scale, we're all aware that closing of borders makes no sense. And we're all aware, of course, that we're complementary. This is the beauty of Europe. Each one of us has, each one of the countries has its own infrastructure. And so some countries might be more able to manufacture, others will maybe be very good in development of vaccines. And I think the idea within the EU has to be to make use of this complementarity and to basically build a big collaborative network around pandemic preparedness and enabling this. I think it is much more complex if we think about the low-income countries, for example, and on the global scale. But we're seeing that there's a lot of initiatives right now to build regional capacity there also for vaccine production. This has two reasons. I think one is that, of course, there might be more trust into the own vaccine production capacity and the skills once this is really built up, and the own, of course, also regulatory capacity to then approve these vaccines. On the other hand, we did see that there was a lot of obstacles also in transport, for example, and distribution of vaccines from where they were produced. Now there's a lot of discussion on equity, and I think this can only be resolved if we also expand manufacturing capacity throughout the world.
One last question, and we may discuss this a little bit. On this WHO list on pathogens to come, I see many well-known pathogens from Africa and other countries, and some of them have arrived now in Southern Europe or even in all over Europe. The two diseases I'm missing on the list is influenza and the next corona pandemic virus to come, right? SARS coronavirus 3. We had coronavirus epidemics since 2002, now 3, right? So roughly every 10 years, we can expect a coronavirus pandemic if the series continues. So why are these common pathogens not on the list?
I think this is very true and a very good point. I think that basically the idea here is that we have experience with these pathogens, but I'm not part of the committee, so I don't really know. And that we do, independently of this list, have the possibility to ensure preparedness. We know basically which antigens we have to choose and how to work with. This was also, I mean, very good with SARS coronavirus 2, that we knew what structures of the virus were important. And so I think that probably this is not considered as high priority because there is more knowns.
Okay. So I guess in the end, research on this is done anyway, and maybe you don't need WHO support for additional funding or initiatives to get this done. Yeah. Very interesting. I have no further comments or no further questions, actually. Maybe you want to have one last word, a main message you want to share with us?
Yes, thank you very much. Thank you for inviting me today. I would like to share the message that I think that the most important aspect of pandemic preparedness is to collaborate and to communicate. So I'd like to invite everybody to share their knowledge and to contribute to the pandemic preparedness actions wherever you are. Thank you
very much. That was a very nice last word. Openness and transparency and sharing. And we just saw the publication from Wuhan, how it all started with the with the COVID pandemic. And anyway, thank you very much again. It was a great honor and pleasure to have you with us today. This was our Global Healthcare special on June 9, 2023 on pandemic preparedness with our special guest, Professor Bekeredjian-Ding from the German Center for Pandemic Vaccines and Therapeutics. You will find today's slide presentation in our show notes. Please share, like, and leave your comments below. Thanks for joining us today. Goodbye from Germany.